Hello students, this is Dr. M. Bhushanam, Assistant Professor in Zoology, Maharani Science College for Women, Bengaluru. Students, today we are going to learn another topic of developmental biology and organic evolution paper of Zoology 7 that includes 3.2c, the role of evolutionary forces in speciation, and the fourth type is genetic drift. Genetic drift is an evolutionary force that brings in change in the allelic frequency of a population by a random process or a mere chance. It involves natural disaster or the process of migration. Drift means to change that keeps the allelic frequency apart. So allelic frequency will change of a particular population. It is due to random chances that includes natural disasters or migration processes. The primary condition required here for genetic drift is population should be smaller. The effects will be very strong. In genetic drift, there is overall shifting of allele distribution in a separated small population. The concept of genetic drift was given by Sewall Wright in 1929. He defines genetic drift as the random directionless change in allele frequency that occurs significantly in smaller population. Students, let me explain the definition here now. In a small population of a particular geographical area will show sudden change in the frequency of alleles randomly. So this change in allele frequency of a small population is due to two important conditions. Condition number one is natural disasters like earthquakes, volcanoes, cyclones, etc. Any natural disasters. Number two, migration. So when these forces act on a small population, the frequency of alleles will change. This we call it as genetic drift, and it was given by Sewall Wright. Down in the slide we can see two groups of insects. In first group, which is considered as the original population, there are two forms of insects, one of red, the other of green. Now due to migrational force, few red variety of insects move to an isolated place by mere chance and do not come back. So they settle and colonize in the new migrated place, show certain of changes in their alleles because they need to live in a newer environment. There they survive and reproduce. Hence, in this new place, there are no green varieties that we can find. So newly migrated colony has no frequency of green color alleles at all. Sorry, green color related alleles, yeah. But shows increase of red color related alleles. So the frequency of red and green alleles of original population are different from that of newly migrated current population. So this change in allele frequency of a smaller population which takes place merely by chance is called genetic drift. I think the concept is clear. So when you compare to allelic frequency of the original population with that of the current population, green color related alleles are totally absent. So you find only red color alleles. So there is totally allelic frequency change compared to the original population and the migrated population. This is called genetic drift. The new allele, if causes more reproduction of DNA, obviously that new allele increases in its number and the frequency of that new allele is also more, or vice versa process. If it is beneficial to an organism, let us call it as positive selection pressure. If it is harmful, we call it as negative selection pressure. So positive selection pressure brings in genetic variations which are helpful to the organism. Negative selection pressure will bring in harmful genetic allelic frequency in them. How this allelic frequency with within a population will show the change. Here is an example to cite. If we have a troop of gorilla, so gorilla lives in groups, troop, where you find hierarchy. Let's consider there is a big power male, strong male. It will mate with more or all females of the troop, for it is being very strong. And when that male alone will mate with all the females, the condition what we find here is his genes will be passed on to the next generations. So he is the leader or dominant of the troop, and rest all males will be suppressed from the process of reproduction. So he will be given much of chance to mate with all the females. So his genes will be passed down in the next generations. He will inherit his genes into the offsprings. So leader genes will become much seen in the troop. And now this is what is happening here. And you remember, students, leader gorilla's genes when are inherited, but not the rest of male gorillas of the troop. Strong ones are selected by the nature. And we call this process as natural selection. When strong male genes when are inherited to all the offsprings of the troop, rest of all the males will be suppressed there. So we call it as natural selection. Now comes the concept of genetic drift. In genetic drift, there occurs an external force of natural disaster. So what is genetic drift? It is the change in allelic frequency due to random chance of forces like natural disasters or migration in smaller population. So that's the definition, right? So in genetic drift, what happens is that There occurs a natural disaster like a forest fire or hunting or earthquake, any of these chances in which the strongest gorilla dies or killed. The strong gorilla of the troop is killed now. Now, to have the process of reproduction, the other male will be available. So, the other male's genes now are passed on to the generations. So, this male will have the other males will have the chance to mate with the rest of females. So, new individual genes are now passed on to generations, and it is purely by choice or randomness. Students, remember, since male gorilla was strong in the earlier case, the natural disaster did not leave it or select it. Nature do not care for the strength of male gorilla. So, it just killed it. Now, the weak male of the troop is given the chance to mate. So, it is selected by the nature. So, this is what we call it as random selection by nature. So, new individual genes are now passed on into generations. So, what's happening to that allelic frequency of the original population and the current population? It totally changes. Now, the strongest male gorilla presence is not there. New gorilla, male gorilla's genes are passed on. So, new population, new individuals of the population are seen. Weaker one is selected. So, this kind of changes that we find in the allelic frequency due to mere chance of external forces. We call it as genetic drift. The next requirement of genetic drift is small population. Large population do not show much of changes. For example, if one individual of 10 population died at a younger age, before passing its genes into next generation, 1/10 of alleles of gene pool are suddenly lost. Correct? Of 10 individuals, one before mating is dead. It is killed otherwise. So 1/10 of the gene pool frequency is lost. It is like if we have 10 rupees with us, and if we lose 1 rupee, the loss is much significant. So 1 rupee out of 10 rupees, when is lost, leaving behind the 9 rupees, it matters more when compared to if you have 100 individuals in a population and one individual dies. 1% of the genic allele is lost. So the change is not that very significant. I mean, in a second case, if you have 100 rupees with you, if you lose 1 rupee out of it, the change, the loss that you have experienced is less significant when you compare the same 1 rupee out of 100 rupees. But out of 10 rupees, if you lose 1 rupee, that change is more significant. So, smaller population, if shows genetic drift, the variation is very significant. The change in allelic frequency is very significant when we compare it to larger population. So, genetic drift requires very small population. So, random change, small population. Okay, and random change is due to, chance is due to the disasters and the migration process. This you'll have to remember. Just now I explained how exactly the larger population change in allelic frequency shows less significance of the change of alleles, and in a smaller population the allelic frequency change will matter a lot. It's very significant. Now coming on to the types of genetic drift. Students, this concept of genetic drift is very, very important. It is most repeated question in the examinations for short notes. So first you'll have to define what genetic drift is. Then what is its significance? What are the requirements? Next comes the types. There are two types of genetic drift. Number one, bottleneck effect. Number two, founder effect. First one we shall look into is bottleneck effect. In this type of genetic drift, the natural disasters are the major causative factors that kills the organisms, making the original population to lose the alleles, bringing the change in allelic frequency. So natural disasters include examples like volcanic eruptions, earthquakes, then hunting, forest fire, then habitat fragmentation, then forest fire, etc. So here, due to killing of the individuals of population, original population, there is change in allelic frequency. Down in this slide, there is a picture representing bottleneck effect of genetic drift, which is popularly known as bead lab experiment. The left side, first bottle, indicates the original population. So you have the bottle filled with the, totally filled with the population. Students, in the bottle we find three colors of balls, indicating three different types of alleles. One is red, second one is yellow, third one is green. So towards the left side. Now, the natural disaster should act upon the original population so that few are left and few are selected. So for this process to explain, or this step to understand, we need to tilt the bottle upside down into another new bottle. So the neck of the first bottle will allow only few colored balls, irrespective of their colors, fall into the new bottle, new empty bottle by choice. We here the balls that fall into the newer bottle is representing the selected new individuals after the disaster. So by chance, you are allowing few of the balls to fall into the newer bottle. Now, the third sequence of the bottles indicates the selected alleles. Now, these selected alleles show the third sequence shows 3 red balls, 3 green balls, 1 yellow ball. So allele frequency is getting changed for this particular group because in the first extreme left, original population indicates more of yellow, less of red. But in the last towards the right side, when you look at that selected population has multiplied, indicating it is also filling the bottle, but with more of red and green and less of yellow. So now you compare both the extremities of bottles which are filled with the balls. There is color change for the balls, and this is how the allelic frequency will totally change between the original population and the current population. So in between these two, there is the selection process by the natural disaster by means of mere chance or randomness. So this type of allelic frequency where the natural disasters will act upon the original population, kills few and leaves few back, and the left out individuals which are selected by nature now starts multiplying, showing a newer allelic frequency. When we compare this allelic frequency of the original population and the newer population, it totally varies. This change in allelic frequency, we call it as bottleneck effect. Are we able to follow? So bottleneck effect means that bottleneck exactly referring to the natural disaster selection, which is very random. Okay, so this is what we find in the first type of genetic drift. Students, remember here killing of the population, no change in the gene sequence. It will remain same, okay? And that has undergone multiplication. So allelic frequency got changed, no mutation, okay? Here is a case study example for the bottleneck effect of genetic drift. That is northern elephant seal. So this seal's head looks like that of the head of elephant. So we call it as northern elephant seal. In 18th century, their number were 30,000, but in 1890s, the number were just 20 in number. 30,000 number in 90 years of span has reduced to 20. People later thought that hunting was the force which acted upon it. There is drastic reduction in their number. So the large population got reduced to very small. This we call it as bottleneck effect of genetic drift, which was later understood by the scientists. And that new population which was resulted totally showed a different genetic variation. So allelic frequency is entirely different. So in bottleneck effect of genetic drift shows the genetic structure of survival population common for the entire new population is in later generations. In bottleneck effect occurs only when a few individuals survive and reduces variation in the gene pool otherwise. This allelic frequency is very different from that of the original population. The variation of survived population is seen in all new individuals. So totally a new species gets formed. The other external force selected related to bottleneck effect of genetic drift is competition, then predators, diseases, and so on. So remember, the variation of survived population is seen in all the individuals so that a new species gets formed. Here is a slide to show bottleneck effect of genetic drift in elephant seal that we have discussed just now. In case 1 of original population, that is extreme left shows elephant seal of brown, white, grey and black ones. In the second, only more of grey, less brown and white ones were selected by bottleneck effect. In the third set, the selected individuals reproduce, and in the last pick, yellow browns were selected, which are increased by genetic drift. Also, the black ones are totally absent in the extreme right. So this allelic frequency change in the small population that occurs due to randomization or external natural disasters or by migration is called as bottleneck effect. Okay. So after we understanding the first type of genetic drift called bottleneck effect, next type of genetic drift that we are going to understand now is founder effect. Here, the external force is in the form of a barrier or simply by means of migration. Students, in first case, natural disasters will kill out of 10, 1 will be killed, 9 is left. Okay, those 9 individuals will totally change their frequency. When their number gets reduced, they form a newer generation, newer species. Whereas in founder effect, the external force that acts upon is the migration force. We also call it as gene flow. When migration occurs between one group and the other group, or one area to another area, there is transfer of genes from one population to another. Okay, this we will have to remember. So migration is also called as gene flow. Due to external force of migration, few individuals of population will migrate to a newer location. So there are many forces that will make the organism to migrate. So after they migrate to a newer location, they do not return back. They don't come back to the original place. So they get adapted to a newer environment, reproduce, and there they show a newer allelic frequency. But what happened to the original frequency of population of alleles? It is totally different. So in this slide at the top is a picture depicting founder effect. The original population includes yellow, black, white species of flies. But the migrants do not have white ones. It is between yellow and black, right? So only black and yellow ones reproduce in the newer place, excluding the white ones. If we compare the allelic frequency of newer and original population, it is entirely different. This type of allelic frequency change that has led to a newer generation, okay, newer species due to the migrants is called as founder effect. So there is finding of a newer species. Okay, so that's what founder effect talks about. The popular case study includes a founder effect is Amish people with polydactyly condition. Students, remember, Amish people are group of German people with the variance in church origin. So they, when compared to the rest of German people, showed more incident of the genes responsible for polydactyly condition. Poly means many, dactyly means digits. So instead of normally people having 5 digits per limb, they will have more number of digits, as what you find in the picture here. So both for the hands and legs, you find more number of digits. We call it as polydactyly condition. This is due to founder effect of genetic drift. It is because more incidence of this gene responsible for polydactyly is seen in this group of people, not in all the German people. Okay. So only in this group of Amish people we find more incidence of this gene. Next case study example is Huntington's disease in Dutch Afrikaners. Huntington disease is a disease related to degeneration of nerve cells in brain. It was commonly seen in Dutch people who settled at Africa. This race of people are called as Dutch Afrikaners. So they had more incidence of the genes responsible for this disease. Similarly, when two birds of the same species migrate to a newer island, their alleles are responsible for the diversity on that particular island. So few examples of genetic drift are recorded here. American bisons showed endangerment has now recovered in its number, but with a very little genetic variations. So the number got reduced, but people have managed to provide them the mating facilities, and now their number has increased. But the present allelic frequency of these bisons and the original population that was there, when we compare, it is totally different. So this is called genetic drift. Similarly, among rabbit population of brown and white fur, even though white is dominant allelic, it is not selected by nature, but the brown one is selected by the phenomenon called as genetic drift. So there is a rule in Mendelian genetics that dominant should be selected, but here it is not so. Genetic drift acts upon the population. So this is about the importance of genetic drift concepts. Next we have the other example is a couple with the brown and blue eyes has children with brown and blue eyes. Even if there is a chance of 50% for having blue eyes, brown eyes being dominant alleles, all the children will have only brown eyes in future generation. So what happened to that blue eyes? So those alleles are not selected by the nature. By genetic drift, they are lost. Bird has an allele for two different sizes of beaks. So genetic drift might eliminate one of the beak size in the population, bringing in certain genetic variations, and only one gene, one type of beak is selected in future, which is entirely different. It is due to genetic drift. So after we understanding the concept of genetic drift, its types called bottleneck effect and founder's effect, now we shall try to answer the MCQs. Question 1. Genetic drift is the change in allele frequency of a population due to which of this particular options? It is random chance. Answer is A. Genetic drift is more likely to happen in very small population. B. This mechanism of evolution occurs when individuals migrate between populations. So it is gene flow. B. Which of the following is an example of founder effect? Bald eagles are hunted until near extinction. Population recovers. Small group of iguanas floats to an island, starts a new population. Red fox pups are more likely to survive in a snowy environment if they are born white. So founder effect is you find totally a newer population. Okay, so their allelic